So it's my pleasure to introduce the second plenary session. This is the last session of the day. My name is Eric Smith. I'm a professor of neurology at the University of Calgary in Canada. I have no disclosures relevant to this session and topic. Please, if you have questions, we'll have some time for questions at the end. I hope you can put them in the chat and I'll monitor the chat. And now we'll go ahead and introduce our plenary speaker, Dr. Gary Rosenberg, who's professor of neurology at the University of New Mexico and the founder of the Memory and Aging Center there. To me, there's at least two hallmarks of Gary's work. One is the diversity of methods that he applies to the study of small vessel disease, including advanced neuroimaging that many of us are using, but he integrates that with biofluid biomarkers, including CSF. A second hallmark is the care with which he takes to subtype and phenotype small vessel disease. So as we know, to many people outside our field, white spots on MRI are UBOs, but Gary has shown us that they are quite diverse in their pathophysiology and their outcomes. And he's made some seminal discoveries, including early work on the importance of blood-brain barrier permeability and how it evolves over time in small vessel disease. So without further ado, Gary, please go ahead. We're eagerly looking forward to your lecture. Thank you.
Yeah, thank you very much for that introduction, and thank you for the opportunity that the committee has given me to summarize some of the work we've done over the past few years. Also, I appreciate the committee's care in helping me by getting a very good introduction by some people that have done similar sort of work. And also I appreciate the timing. So it's not 3 in the morning in New Mexico, but it's a more reasonable time. Is there any way to get a pointer? I suspect I won't be able to do that from here.
You can take it off from your on the Zoom.
Oh.
Oh no! Oh, actually, no, you can't because you're not controlling it yourself, Gary. Sorry.
Yeah, I thought. Okay. Next slide, please. So what I'm going to talk about is the ability to find mixed dementia patients during life by using 2 biomarkers. One is white matter injury, mainly with diffusion MRI, and the other is CSF proteins for Alzheimer's disease. And then I'm going to talk about similar things that we've heard about multiplex protein assays. And these make very large databases. And so we use random forest to find the biomarkers of what are called variables of importance. And what I'll do is relate them to the breakdown of the matrix, extracellular matrix and the opening of the barrier. And then the important thing, obviously, is to show that these biomarkers relate to cognitive dysfunction. But I'm also going to show you how they relate to albumin index, which I think is more important than I had thought before. Next slide, please. One of the main problems that we started with, as Eric alluded to, was clinically, it's almost impossible to separate some of the patients. The Alzheimer patients look like the vascular patients. The vascular patients look like the Alzheimer patients, which is one of the reasons we started looking at biomarkers. But what it turns out as time has gone on is that the most important group is the mixed dementia group. And we don't have good ways of sort of detecting those. And what I'm going to show you is that we can make that diagnosis during life by using a set of biomarkers, multimodal biomarkers. And then I'm going to sort of relate the diffusion MRI to the subcortical ischemic vascular disease, which is the one that's the small vessel disease and the one that we've been focused on. Next slide, please. So we've been sort of misled by the pathology up till recently because particularly the Alzheimer group excluded vascular disease. So we didn't have a cohort with both Alzheimer's and vascular. But that was a mistake. And when Trojanowski and his group with the Alzheimer's cohort went back and looked at it carefully in a paper that they published, Toledo published in Brain, you can see the red is the prevalence of vascular disease in the patients with Alzheimer's disease. And it turns out that maybe 70% of patients with Alzheimer's disease also have vascular disease. And that was not appreciated. Next slide. And a big step was made when the Alzheimer's disease has been way ahead of us in terms of biomarkers. They started quite early, and they've had amyloid, tau, and so-called neurodegeneration. And if you're a clinical person, you also include cognition. And then Jack and his colleagues came up with the biological definition of Alzheimer's disease, the ATN formula. A is amyloid, and you can get that either with PET or CSF. And T is tau, and that's either PET or CSF. And neurodegeneration is loss of tissue. Next slide, please. And this is the sort of thing that the Alzheimer world has been looking at for a long time, and we are just beginning as a vascular group to look at it. And you can see in the left, the patient with the amyloid, tau, and neurodegeneration, everything is positive. And then there's some patients that don't have the tau, but have a lot of amyloid and have the degeneration. And then there's some that don't have tau or neurodegeneration, but just have amyloid. So there's all these different combinations. Next slide, please. So what we have been looking at, and this is part of the NIH consortium, the MARC-VCID consortium, we're part of that, is looking at the vascular component of Alzheimer's disease. So the question here, and this is a question that I started looking at a long time ago, is how do we use FLAIR? FLAIR is so nonspecific, and it shows a lot of normal people that if you try to rely on FLAIR, you're going to be overdiagnosing this disease. And that's what they did in the early days. A lot of people were so-called Binswanger patients. And of course, they weren't pathological. So when you go beyond the FLAIR and use diffusion tensor imaging, and now in the MARC-VCID group, we're looking at free water, which is even better, as the marker of white matter injury. And then of course, there's a lot of biochemical fluid markers. So we think the formula should be ATNV if we're gonna really look at people in a more broader sense. Next slide. So when we started thinking about this, there's really two ways that you can get to mixed dementia. And one is through the pure Alzheimer's disease pathway, starting with amyloid, tau, neuronal death, et cetera, et cetera. And that usually can begin as early as 40. And by the time you're 70, you'll have all the factors. There's also a pure SIVD pathway in people that have severe hypertension in their 40s. By the time they get to 60, they have inflammation, blood-brain barrier opening, white matter damage. But there's many patients who fall into both categories. And what do we do with those? Next slide, please. So we've taken the approach that it's going to be hard to classify people into 4 groups: Alzheimer's disease, mixed, leukoariosis, and SIVD, unless you break it down into 2 axes. And this is what Avram Kaprayan has called the double dichotomy, which allows us to pick out the mixed dementia patients. So on the y-axis is the Alzheimer's disease severity score. That's the proteins in the spinal fluid, and it could be other factors also. And on the x-axis is the vascular damage, as shown primarily by DTI. And we've used this to classify patients into these 4 groups. The leukoariosis group has been the challenge. These are people with flare white matter damage, but not much in the way of DTI damage, and their cognition is not impaired. So this is an important group to eliminate if you're going to, particularly if you're going to do clinical trials. You don't want to have a big group with no cognitive problems, and you're following the white matter. And this is our data. This is the UNM MARC VCID cohort. And you can see in the left upper quadrant are the Alzheimer patients. These are high ADS score and not much VDS score. And if you move over, you'll see the mixed patients, the green. And in the lower quadrant on the left is the patients with leukoariosis, no cognitive problems. And then in the lower right quadrant is the SIVD patients. And then there are obviously going to be people on the line, as you can see here. So it's not perfect, but it gives you an idea of how to use cluster analysis. And these are two clusters, one cluster analyzing the Alzheimer proteins and one cluster analyzing the DTI score. So I think this is a process that will be similar to the Alzheimer biological diagnosis, but by adding in the V. And actually in their paper, they had said there should have been a V, or there'll be other things too, probably. Next slide. And this is the paradigm that I proposed some years ago, which seems to be holding up, that these patients have hypotension, hypoperfusion, not hypotension necessarily, due to the damage to the blood vessels, the fibrosis, the hyalinosis. And they become hypoxic, and hypoxia induces the hypoxia-inducible factor 1 alpha, and that creates this cassette of genes. On one end, it's an injury pathway with the convertase FUR gene leading to the furin molecule, and that activates the membrane type metalloproteinase, which in turn activates MMP2, which is the intrinsic MMP, and that leads to blood-brain barrier breakdown, endothelin-1 activation, and from that you get edema. And also the MMPs attack the myelin, which gives you the axonal damage. So it's the perfect model for the white matter disease. But on the other hand, you have repair, and this is where angiogenesis comes in. And that complicates things because the metalloproteinases are also involved in sprouting and other forms of angiogenesis. Next slide, please. So this is the model that Dr. Adicola came up with for the inflammatory disruption of the blood-brain barrier, and he's included the metalloproteinases, the reactive oxygen species, and the cytokines. So hypoxia, of course, induces all of these factors, and exactly how they work, we're just beginning to learn. Probably the metalloproteinases are breaking the tight junctions and damaging the extracellular matrix around the basal lamina, around the blood vessel. And then you have the cytokines coming in and aggravating or perpetuating the inflammation. And this is where these multiplex assays have been helpful. Of course, there's other things going on, as you can see from the diagram. Next slide, please. I'm going to show you some of the sort of thing that Marcus is doing. This is how the blood-brain barrier is disrupted in our Binswanger patients. And the Binswanger patients are the ones with the extensive small vessel disease. The flare image is very dramatic. And the dynamic contrast enhanced MRI, which is on the right, the green is the white matter lesion. The red is an early blood-brain barrier opening, and the yellow is a severe blood-brain barrier opening. And you can see that it's the white matter is the primary site. And this is what we saw early on when we started doing this. Next slide. This is a study that was done by Branko Wisa when he was with us before he went to San Diego. And what he and Arvind Kaprahan did was put a 4 millimeter rim around the white matter lesion and look at the relationship of the penumbra to the injury. And here you can see that the normal appearing white matter is the place that seems to be getting the most injury. And about 52% was in the rim. Next slide. So this is a little different tack now. This is the inflammatory molecules that we've been looking at. So the MARC-VCID is looking at proteases, angiogenic growth factor, and cytokines. These are primarily measured with mesoscale discovery. Now that Quanterix is around, we're switching to some of that. But we had the ability to look at 5 of the matrix metalloproteinases. Everybody looked at 2 and 9 because that was the zymogram gelatinases, but now we can look at a much broader spectrum. There's a group of angiogenic growth factors, VEGF, FLT, TIE, placental growth factor, and then there's this long list of cytokines. Next slide. And I just wanted to show you, you can't really see the heat map, but this is all of the biomarkers we looked at correlated with cognition. And on the right, you can see the summary. The memory function correlated with MMP10, VEGF-C, IL-2, TNF-alpha in the CSF, IL-6 and TIE2 in the plasma. Plasma turns out to be not very helpful in this analysis. CSF is really the key. Executive dysfunction was found to correlate with MMP1, IL-1 beta, and IL-13, and in the plasma, MMP2. And this was just recently accepted for publication. Next slide. So I've always been thinking that the gadolinium was the key, but we also, because we were doing MS studies in the beginning, we were trying to rule out MS in our white matter patients. We had albumin index. And when I went and ran the albumin index, it turned out that it correlates with MMP2, MMP3, MMP10. And that's in this large group of dementia patients. And you can see that there's pretty good correlation with log-log transformation to take out the outliers. Next slide. But several other things also correlated with the albumin index besides the proteases, and that was VEGF-C, FLT1, placental growth factor, and the cytokines that you've heard about, IL-1 beta, IL-6, IL-8, and TNF-alpha. So this is nice correlation with the talks that you heard earlier. Next slide. So the problem is, why are we getting differences with albumin and with gadolinium? And there's some very nice studies that were actually done some years ago on hypertensive and acute hypertensive animals with electron microscopy. This was done by Surinette Nang when she was working in Canada. And you can see in this, it's not HRP, but electron micrograph of the lanthanum, that there's a lot of pinocytosis. And what they showed was that the arteriolar, not the capillary, was having pinocytosis. So how does a molecule get across the blood-brain barrier? It can go through the capillary by through the blood vessel through pinocytosis, or it can go across the tight junctions. Hypertension leads to the pinocytosis, and that's why albumin turns out to be in the spinal fluid in vascular dementia. And the gadolinium, which is leaking in inflammation, multiple sclerosis, probably is going through tight junctions, because you don't get a lot of albumin in the MS patients, but you get that in the vascular dementia patients. New slide, please. Next slide. So what I've tried to tell you in the time I've had is that we can now pick out the Alzheimer patients with Alzheimer proteins, the CSIVB, the subcortical ischemic vascular disease, small vessel patients with the MRI DTI. And by putting those two together, we can come up with a group called the mixed dementia. And I think a lot of the studies that were done earlier that gave confusing results probably didn't separate out these patients as carefully as they should have. So we have now the same data that a lot of people are getting from these multiplex assay kits. And it turns out that several MMPs, several angiogenic factors, and many cytokines are important. And the way you need to pick these out is with some machine learning type approach, because there's too many variables. And the biomarkers actually do correlate to the neuropsychological test results, indicating that they are biologically significant. And finally, I think or at least I didn't pay as much attention to the albumin index as I probably should have, because the albumin index correlates with the MMPs and really shows this proteolytic breakdown of the blood-brain barrier that we've been talking about in animals, but now in people. And I think what we're going to find is that this is a separate mechanism from the opening of the tight junctions, probably be some kind of pinocytotic process. Next slide, please. I've been fortunate to have a very good group of MRI people and biostatisticians and chemistry people, and I've been supported by the Mark VCID, and now we're one of the new ADRCs. NIH created 4 of them, and we're one of them. So this is the group that I work with. Thank you all.
Thank you so much, Gary. That was a great talk. Congratulations on becoming an ADRC. That's a big deal, I know. So that's fantastic. You can raise your hand or put some questions in the chat. Maybe I'll start with a question, and we have like 4 minutes for questions. You mentioned in passing repair, which intrigued me, right? Because this is a big deal in MS. For the most part, people have treated white matter lesions and small vessel disease as irreversible progressive lesions, but I think there's increasing evidence that that's not entirely the case. Like some work by Joanne and others has suggested that some of them may resolve. Now, you've done very careful voxel-based analysis of white matter intensity progression. I was wondering what you thought about the reversibility of white matter hyperintensities? Is it achievable? Is it and is enhancing repair potentially a future mechanism for therapy?
Yeah, I definitely think it is. The problem is that there's not enough longitudinal data. We don't have, that's what this, in fact, the Mark VCID hopefully will go into Mark VCID 2 with the idea of increasing the longitudinal data. I think many people have seen these lesions not persist. And I think what you Marcus and John O'Brien may be seeing is this transient hypoxia. What happens when someone has sleep apnea? They're intermittently hypoxic. That's been very well worked out by the sleep apnea group, and that induces factors that lead to white matter damage, but then they go away. And I think I have some patients now, we haven't measured them as carefully as we probably should, who have had bad sleep apnea, hypertension, and when we finally got everything under control, they seem to stabilize. So I think there's going to be a lot of interest in that part of the story. And how the angiogenic factors fit into that is going to be even more complicated because they leak. The angiogenic vessels leak in the beginning, and then, of course, they stop leaking and they grow into new vessels. So is that good or bad? We're going to have a lot of work to find out about that. That was a great question.
Thank you, Gary. And then we have a question in the chat, and of course my Zoom is like cutting off the name of the speaker. I guess it's Dr. Garanmaye. Sorry, I'm butchering your name, I'm sure. Fatima, sorry. Fatima, did you want to say your question out loud?
Hi, thank you so much, Gary. So I just wondered, where does amyloid angiopathy fit into this classification that you discussed in terms of mixed dementias, both in terms of a classic amyloid angiopathy, but also in the case, let's say somebody has clinical features and imaging features that supports amyloid angiopathy with multiple lobe or microhemorrhages, but an amyloid PET negative, suggesting there's no parenchymal amyloid.
Yeah, actually, Steve Greenberg wrote a very nice paper recently on the difference between the amyloid in the brain and the amyloid in the vessels. I mean, Eric is the expert on that. To be honest with you, I have never understood this conundrum of why amyloid shows up in one place and not in another, and how it affects. Amyloid itself turns on the metalloproteinases. It's an activator for the metalloproteinases. So that leads to bleeding. That's pretty well established. But this, what is the relationship between the Alzheimer protein in the brain and the blood vessel? I mean, you can read Steve Greenberg's paper. I'm not sure I understand that. Maybe Eric has some thoughts on that. I know you've been living with that for a while.
Just to be clear, I don't have amyloid angiopathy, but I've been living it in my research life. Yeah, it's not entirely clear. I mean, it's felt there's a neuronal origin of amyloid, the APPs, transmembrane protein, and then bulk movement of amyloid through the CSF and cleared through different routes, including the IPAD pathway, intramural periarterial drainage. And Roxanne Carrara has done a lot of work on this, for example. And then in some people, it's sticking in the vessel though, right? As it's supposed to be kind of like moving along the basement membrane and out of the brain. And exactly why that is not clear. There is relatively more A beta 40, in the vessel compared to in the brain. But I still feel like we see this clinical spectrum though, right? Where some people are getting the arterial amyloid, loss of vascular integrity for the reasons that Gary says, and then bleeding, and other people predominantly this parenchymal amyloid and tau problems. So Why it's sticking in the vessels in some people is still not entirely clear to me, other than ApoE genotype status probably also involved.